thank you for joining us today. I am Dr. Mo Al-Khalaf, a postdoctoral research fellow at the University of Ottawa Heart Institute, and I am back as your host for today's episode of BEaTS Research Radio, a YouTube channel and podcast streamed normally from an active lab at the Ottawa Heart Institute. Our goal at BEaTS Research Radio is to communicate science to the community. Our guest today is Dr. Elisabeth Bik. Dr. Bik received her PhD in microbiology at Utrecht University in the Netherlands, after which she worked for many years in the School of Medicine at Stanford University researching microbiomes. Dr. Bik now holds the title of consultant and image forensics detective who left her paid job in the industry to search for and report manipulated and falsified images in biomedical articles. Her efforts towards investigating scientific misconduct has contributed to identifying many ways in which some research showed signs of manipulation and falsification. Thank you so much for joining us today, Dr. Bik.
Thank you. I'm glad to be here.
Absolutely. Before we delve into the methods and data, Dr. Bik, I'd like to start by asking you, how did you find your way into this journey of scientific detective work, which is a highly valuable but an atypical path for many scientists to pursue?
Yes, it started as a strange hobby. Basically, I heard or read, and I forgot exactly the occasion, but I read about science misconduct, and I was immediately grabbed by that because there's a lot of juicy stories about people, cheating in science. And as a scientist, I was a little bit appalled, but also very intrigued by it. And so I just started by looking for plagiarism because I heard that was a thing, and that's not allowed in science. And so I started searching for that by putting one of the sentences I had written in a review article in Google Scholar between quotes. And immediately I found a paper that was not mine and that had stolen my sentences. And so I was angry, and I investigated that a little bit further. And this became my new hobby. And I did this day and night after, I worked at Stanford at the time. And I just, as soon as I got home and I got some free time, instead of watching a Netflix, series, I did this. I searched for plagiarism, but also I found out that I could recognize duplicated images. So that sort of took over. But I did this since 2014, and now I quit my job and I am doing this full time.
Wow, that's thank you so much. That's very illuminating, and of course, a valuable skill set. Now let's tackle some of the science. Your investigative focus is biology and biomedical research. Mm-hmm. Is there an overall pattern that emerged when you compared the various types of manipulations you flagged? Do you see a specific method or specific type of research that is more prone to manipulation?
Well, I focus on looking at photos because for the human eye-brain combination, and I scan for these things by eye, photos are fairly easy to recognize manipulations in. If there's manipulation or fabrication, so completely making up data in a table or a line graph, I would not usually be able to recognize that, because anybody can make a line graph with fake numbers and it looks fantastic, and who would know that this experiment is not real? So it's hard for me to find those things. I'm really only finding the really dumb examples where the cheaters left something for me to find. And so I look at duplicated photos, either a simple duplication or a shifted duplication, or mirrored or rotated, or manipulated photos themselves where you see duplicated parts, like maybe the same cell or the same group of cells or the same parts of the tissue is visible multiple times within the same photo. So because I'm focused on photos, that sort of limits most of my searches to biomedical papers, which will have photos of tissues or protein blots and things like that. A lot of papers in chemistry or physics might not have photos. But I've also found manipulated or duplicated photos in physics papers, especially those with nanoparticles when they have scanning electron microscope photos. Some of those are manipulated or duplicated across different papers.
Yeah, and this actually highlights one of the facts, like as a bench scientist myself, I do value the importance of imaging
Hmm.
in an article, that because like you said, it is maybe more difficult to spot any changes in a graph or a table, while in an image, especially like when you see the real image, there is like a stronger proof to the concept and the data that that's being discussed in an imaging type of result figure, as opposed to just showing bar graphs and tables.
That's right. Yeah, and those figures, those photos are part of the data. They are data. It's because sometimes I hear when I mark these things, I hear the reply from the authors along the lines of, "Yeah, but you know, it's only a photo, and our experiments are still true." And I'm like, "No way. This was figure one in your paper. This was showing, I don't know, the structure of your nanoparticles or the structure of your tissues. It's the data, and if that's falsified, then I cannot trust the rest of the data anymore either."
No, absolutely, absolutely. Anything that goes into an article has to be data, otherwise,
Right.
in your figures, otherwise,
Hmm.
and you cannot say something like, this is just a representation, and then I have another one. Well, you should have showed the right representation then.
Yeah,
Yeah.
yeah, I get that a lot where people, when I post these things, and I usually post them on PubPeer, which is a website, pubpeer.com, where you can comment on papers. So when I post things, sometimes authors come in and say, "Oh, just kidding. Here's another photo." But for me, I usually compare that to a doping test in sports, where you just won the race. You just publish your paper, and you have a positive doping test. Your urine, contains some forbidden drug in cycling or whatever sport you're doing. And you cannot just get away with 2 weeks later sending in another drug test that now is negative, right? It's the drug test that counts after winning the race. So if you come up with a photo later that does not show an overlap, for me it doesn't really prove anything other than that you're now a little bit more smarter. You gave me a photo that I cannot find a duplication in, but it's a photo you send in with your paper that I'm looking at, and that one I found a problem in and I cannot trust anymore.
That's a perfect analogy there with the sports. Thank you
Mhm.
so much for sharing it. That's fascinating. Now, let's go on to the next question here. The research context that you investigate has normally passed peer review, the gold standard in science when it comes to published work. In what way can your experience and acts modify and optimize the peer review process so that we can regain this top shelf hygiene and validity of published content?
Yeah, it's a good question, and not easy to answer. Most peer reviewers are not really primed towards looking for misconduct. I think science as a whole is still based on trust. That the manuscript we have in front of us that we peer review is really what happened. And I think most of us, except for myself, are not really engaging any interaction with the manuscript as from the fact that it could be falsified. So I do that obviously because of my work, but most people still think, okay, this is what it is. So one of the things I do to prime people to being a little bit more vigilant for these things is posting manipulated or duplicated images on Twitter. So I have a Twitter account and I post things. I have a hashtags image forensics, and I post a duplicated figure and I say, can you find the duplication? And it's sort of a game because I removed the identifiers. It's not about the paper or the authors, but people now start to see it too, and it's a fun game, but it has a very serious background because I want to prime people to find these things, and I'm getting more and more emails now by people saying, "Oh, I did a peer review and I found one of these duplications now." So that is one way I do it. I also write obviously to the journals and post my concerns on Papier as well, and I feel that slowly publishers and journals are listening, starting to listen to me a little bit, and they're getting a little bit better in rejecting those manuscripts up front or recognizing them. But it's all part of recognizing these things, that it can happen and being aware of it. But the peer reviewer themselves is usually not in a very well trained position to find these things. So it should be more with the publishers and the journals to scan for these things, like they scan for plagiarism in the text as well.
Yeah. And actually, like, this is one of your unique contributions to social media, the idea of crowdsourcing, this way of understanding and analyzing images is actually like, and I've seen it because I'm on Twitter and I follow you. I've seen this for the last couple of years now, and I think it's fantastic that you've managed to basically use the platform of Twitter as a way to crowdsource teaching individuals to become better adept at capturing this type of thing.
Right.
And and I think that, like, if there is a way to make this more of a standardized type of investigative work within the structure of publishing and review, it would only enhance the publication output and content.
That's right, and it's much better for everyone involved if these papers are caught before they get accepted and published, because if a publisher or a journal scans for these things up front and says this is a duplicated image, there might still be an honest error. It might still be caused by an honest error, and so they would give the authors a chance to send in a new photo. But in some cases, it's pretty clear it's an error. I've seen whole figures with 10 different panels that were duplicated. That seems like so obvious that that's some error in uploading the wrong figure twice. But in other cases, they could then reject the paper, and hopefully, of course, they will go somewhere else and try to get it published there. But you give everybody a chance to recognize that it was misconduct or it was an error, and things are not published. Once it's published, once a paper has been published, it's much harder for everybody involved to retract it, and it's a lot of work for the publishers, for the editors, but also for the authors, and not just the authors themselves, but everybody in that lab will now know that their lab has a retracted paper, and it's could damage a lot of people's careers, and there's a lot of other people then who get dragged into these retractions because they're heavily discussed online and might be an embarrassment for everybody involved.
Yeah, no, absolutely. Now, continuing with the discussion about the idea of future directions, what's the plan moving forward to strengthen and implement your methods and efforts on a wider scale, maybe, to contribute to the future of science publication, communication, and even use?
It's there's many people working, for example, on developing software to find these duplications, and that is actually happening right now across different publishers. So there's of course different scientific publishers like Elsevier and Wiley, and there's many of them, and they're now working together in testing software that promises to find these duplications. Because I do it by eye, but I can only scan one paper at a time. And for a human, it's not possible to remember all photos that ever have been published in a scientific paper, while for software, that's doable. And it's computationally maybe intensive, but it's something that I would imagine could happen in the next couple of years. So the software is not that great yet. It has still a lot of false positives and false negatives, but it's getting better. And this is obviously the way to go. Similarly to text similarities, which are found by plagiarism detection software, all manuscripts now in the future, will be scanned for duplicated images. So all images in those papers will be scanned against a database of all images that have ever been published in scientific papers. And that will hopefully prevent people from stealing other people's photos or from having duplicated photos across their own papers. So that's one of the developments that I see will inhibit this. On the other hand, of course, the cheaters will get better too, and they will probably find ways to fly under the radar. Where we've already seen paper mill type of papers where hundreds of papers have very similar looking Western blots, so protein blots, and they're probably artificially generated. So it's we can never keep up with with the
It's an
crooks.
arms
I'm
race,
afraid
isn't it? It's an
it
arms
is. It
race.
is. It's like, but yeah, it's you. Everybody tries the best we can, and I think the trick is obviously to make it a little bit harder for people to cheat. And cheaters are going to cheat. That's not going to go away. That happens not only in science but in any field you can think of. And you just want to make it a little bit harder for them to have
Yeah,
success.
and I think that, like, part of the future is exactly like you mentioned. Well, there'll be some, artificial intelligence, machine learning
Hmm.
angle, probably in hybrid with, human fact checkers afterwards. Post, maybe you require a double
Right.
analysis from one angle, from the computerized, and then double checked by somebody that maybe can have at least half of the abilities that you have in terms of being able to detect, which is not this is a skill on its own, being able to have the visual acuity and ability to detect the kind of changes. Because I tried to do some of your Twitter exercises, and I can't catch half of it. But I do see some that are unfortunately, quite well, maybe fortunately, very easily detected, and
Mhm.
it's surprising that somebody would even attempt doing something like that.
Yeah, some of them I'm looking at these images and I'm like, how do you think we don't see this?
Yeah.
But all the papers I post online have been peer reviewed and have been, seen and sometimes cited by dozens of other papers. And it seems so obviously once you point it out, but other people either didn't see it or did not want to see it. I don't know, but some of these examples are really hilarious. Yeah.
Yeah, which reminds us that actually even it's on us, the readers and consumers of the science, to make sure on our own, without having just the detectives and the algorithms do the work for us. Sometimes we not sometimes we always have to do the work ourselves, making sure that what we're reading, and especially if we're citing it, is as top shelf as possible, and
Mhm.
never skip looking at a figure and just look at, like, read the
Right.
results. Always
Right.
look at the figures before even reading the figure legends. You want to make sure that what you're looking at and what you might be future citing is something that went through your own post publication review by
Right.
yourself, right?
Right, yes. And it only takes seconds, at least for me, to see these things. So, in a peer review, that might take hours. Mine usually take, 4 or 5 hours or so to do. It would only add a couple of extra seconds to look at these images. Once you've seen some examples, they become easy to recognize. So that's what I'm hoping to accomplish by posting these things on Twitter.
These are great. These are great points, and definitely a worthwhile pursuit for all of
Mhm.
us. So, our last question today is: what type of advice or words of wisdom would you like to share with either early career scientists or the wider science-consuming public, in a way to elevate their standards and care for the scientific process and the vital contribution it provides our society?
Right. So basically, two questions. So for early career scientists, uh, I would say don't cheat, and that sounds very easy. But I know that some people are in a situation where cheating appears to be the only way out. When you're in a lab and you maybe are on a visa and you want to have that one paper that will be your ticket to a career, it seems the obvious choice that if cheating, which doesn't have a lot of punishment, right? There's not a lot of punishment for cheating in science. Cheating might actually be your ticket out and might be the paper you wanted and the step up in your career. So I can totally understand how difficult the situation must be, how sad the stories are behind
The
misconduct, and
temptation.
yeah, exactly. And everybody's in that situation, not early career, but also late career. If you're a professor and you need to have your quotum of, X number of papers published that year, or X number of graduate students um, getting their PhD that year, we're all in a situation where we're held accountable for the number of publications. But eventually it will catch up with you and you'll be caught and it will be the end of your career. So don't do it. But it's easier said than done. And for science as a whole, I'm actually very concerned because I feel that all the cheating in science is very damaging to the image that other people will have from science. Like the general audience, by hearing about science misconduct, might actually start to think that science is not to be trusted. While we, that's not what it is. I still firmly believe in science, and I feel science is looking for the truth, and we should not have anybody cheating in while doing that. And because it's not only damaging for the scientist who's trying to replicate these results, it's damaging for science as a whole. And there's already a lot of people who don't believe in science, and that number is rising. And I feel that these cases of misconduct are only helping more and more people move to that side, to not believe science. And
Negative contributions.
yeah, and I hope that people also realize there's people like me, and I'm not alone. There's many other people doing this work, but they're more anonymous. We are a group of people trying to correct that. We're trying to fight for science. We all do this as volunteers, and we try to break through this cycle of conflict of interest where institutes and publishers are not willing to change because they're more interested in selling publications than selling good science. And we're hoping to break through that and change science a little bit for the better.
That's a hopeful note to end this conversation.
Mm-hmm.
This concludes our interview. Once again, I'd like to thank our guest, Dr. Elisabeth Bik, for the valuable effort contributing to the elevation of science quality and dissemination. And of course, thanks for joining us today on this episode of BEaTS Research Radio and sharing some of your knowledge with us today, Dr. Bik.
You're welcome.
And this concludes our interview. BEaTS Research Radio is supported by the University of Ottawa Heart Institute, BEaTS Research Lab, Department of Biochemistry, Microbiology, and Immunology in the Faculty of Medicine at the University of Ottawa. Our AV producer is Alex Ross, and our director is Dr. Emilio Alarcon. Thank you all for tuning in. Don't forget to subscribe to our YouTube channel and our podcast channels. And we'll soon see you again on another Science for the Community episode.